So this was a randomized trial in over 900 patients, in patients who had resected high-risk melanoma. That is, they had their tumors surgically removed. They had stage 3B, 3C, or 4 melanoma, which means locally advanced or metastatic melanoma, surgically removed, rendered free of disease, but over 5 years, they were all at least a 50% risk of return of the tumor or relapse, and over 10 years, at least a 50% risk of death. So that's our definition of a high-risk patient. And these folks were randomly allocated 1-to-1 to receive either nivolumab, which is a PD-1 blocking antibody that has activity in metastatic melanoma and is approved around the world for that indication, or ipilimumab, which is a different type of checkpoint inhibitory antibody that blocks something called CTLA-4, and that is approved in the US as adjuvant therapy and also for metastatic disease, and has been approved at least 6 years for metastatic disease and was approved almost 2 years ago in the US, but not in the EU or Canada or any other countries as adjuvant therapy. So this was a 1-to-1 head-to-head randomized trial of nivolumab versus the standard ipilimumab. And the results of that study were that there was a clinically and very statistically significant benefit for the nivolumab group compared to the ipilimumab group. Relapse-free survival was the primary endpoint, and the study was stopped early at an interim analysis, which is always a good sign. So it had a planned interim analysis, and it was stopped because the Data Safety Monitoring Committee reported the results and felt that it was inappropriate to continue. So the study was unblinded, and clear benefit for the nivolumab group with a hazard ratio of 0.65, meaning a 35% reduction in the risk of relapse over time, and a significant decrease in the risk of distant metastasis-free survival, which was not a primary endpoint. It was a post hoc analysis, and nivolumab was considerably less toxic than ipilimumab. The rate of, say, grade 3-4 significant immune adverse events was only 9% in the nivolumab group and more than 40% in the group that got ipilimumab. And if you look at the most important parameter to me as the practitioner, which is how often did you have to stop because of toxicity in either group, it was less than 5% in nivolumab, and it was more than 30% with ipilimumab, which is consistent with the experience of people in metastatic disease, suggesting that nivolumab was a much better tolerated drug. So you can't lose. Nivolumab is a much more effective drug than an active control of ipilimumab, and it's less toxic. So we hope that it will get registered eventually in the United States. I assume that's in progress, and I suspect eventually it will get registered around the world, and it will become the standard adjuvant therapy for high-risk resected melanoma. Well, I think the death of CTLA-4 is premature, or reports of its death are not accurate. There recently was reported a combination trial of ipi-nivo and renal cell cancer that's quite positive. We'll be hearing some of those data, I think, today. There is no question that in melanoma, the survival of patients who get ipi with nivo or ipilimumab with nivolumab at year 2 and year 3 compared to nivolumab alone seems to be higher, especially in the PD-L1 negative group. In, for example, patients who get adjuvant nivolumab therapy in the future, what will you do if they relapse while on treatment or within 6 months of being treated with their adjuvant nivolumab? You're probably going to want to give them adjuvant ipilimumab. And because of the results we heard at ASCO, it looks like 3 milligrams per kilogram of ipilimumab with respect to relapse-free survival benefit looks as good as 10 milligrams per kilogram. 3 milligrams is less toxic. That's more palatable for patients. So I think the reports of ipilimumab's demise are premature. And I think it goes the other way. Since everybody from now on who has stage 3 or 4 resected melanoma will be nivolumab experienced, it'll change the biology of the relapsing patients, and many of them may not benefit from just more nivolumab in metastatic disease. I think it'll open up new opportunities for the use of ipilimumab. I think ipi plus nivo will have activity in lung cancer. Now it's got activity in kidney cancer. We know it has durable benefit in melanoma. So I think if we can attenuate the toxicity somewhat by changing the dose and schedule, I think there'll be a significant place for ipilimumab in the cancer armamentarium going forward. Well, it depends on your point of view. In the clinical oncology field, you can be a glass half empty person or a glass half full person. I may be a cynic by reputation and by nature, and a skeptic because I was raised and educated as a scientist, but I would agree that ipilimumab can have benefit, certainly in melanoma as a single agent and in combination with PD-1 blockade. And let's say some of the new pembrolizumab epacadostat or pembrolizumab TVEC trials are positive, and let's say people aren't using ipi plus nivo anymore. What do you do when you fail pembro epacadostat? Then you're going to want to move quickly to an ipilimumab combination. So again, it opens up new possibilities of changing the dose and schedule of ipi and adding another drug, which may boost its activity, or it may encourage the development of drugs that target ipilimumab to the tumor microenvironment, or allow it to only act in the tumor microenvironment, which would make it less toxic. Well, there are, and I should give due disclosure, I work with a company that makes a masked CTLA-4 antibody, but that antibody, I think, has promise. There are other antibodies that people are trying to target to the tumor. I think that they would have considerable benefit if they're developed properly.